Professor Craig Banks. Today I'll be chairing the session with René. So René's given me some blurb to speak about and his prestigious career, so I'll talk through that and tell you what I think of the man. So René has over 20 years of experience of research and teaching in computer science, computational modelling and complex systems in Europe and US institutions, with 9 years of various startup companies in the software industry. Alumni of ENS in Paris, a very prestigious place, completed his PhD in 1991 and a postdoc in neural networks in Germany. He's the former director of the Complex Systems Institute in Paris, secretary of the International Society for Artificial Life Systems. He's also co-founded and coordinated the European Erasmus Mundus Masters in Complex System Science. That's called Polytechnic Paris, where he was guest lecturer for many years. His main themes are agent-based simulations of natural morphogenesis, which I hope you can explain to us what this topic is about today.
Yes,
yeah, absolutely. Basically, René's published around about 56 papers, 12 book chapters preceding special issues, etc., workshops, conferences, most importantly 22 postdocs, PhD and MSc students. He's got a H index of 16, which is very competitive for his area. Nearly over 4,000 citations with the Google Scholar. He's got many papers in Nature Communications, which he hasn't written down on here. I'm going to do some digging around. ACS Synthetic Biology. I know Martin will like that journal. So René is a very gentle and modest prof, okay, who brings exceptional expertise and is internationally leading. Okay. Quite simply, we need more profs like René in this faculty. I think that introduces you.
Well, thank you very much, Craig. Thank you very much, Craig, for this introduction, and I must make clear that those last two sentences were not for me. I did not put them in his mouth. And thank you to Amy and the team, so Natasha, who contacted me earlier this year, and Karen, so from the Enterprise team, for organizing this event. I'm very glad and honored to be, I think, the first of this new academic year. So they kind of managed to convince me to overcome my natural shyness and present you with some of my research topics. I hope entertain you with them tonight for 45 minutes, not one more. So those topics are what? They are namely, in put in short, computer models of biology and biological models of computing. So both sides, and how those two sides can in fact collaborate, how we can establish bridges between trying to understand natural systems, especially biological systems, and create new technology inspired by our understanding of those systems. So I'm going to talk about this tonight, and I will illustrate those ideas with several studies, maybe too many, I might skip one or two depending on the time, that I have conducted or collaborated with over the years. So anyway, I must also add a word that I'm so happy and excited to be here at Manchester Met. I joined earlier this year, January, and it's really fantastic. I mean, it's an environment that I feel is very keen on fostering research, new ideas, original work, and I very much look forward building interesting projects with colleagues. Several of them are here in the room. So I'm really happy to be here after a very complex career path. So Craig asked me to say maybe 2 words about that, but I really won't. So you can see here visually a few of the institutions I come from. Lately from France, obviously Paris, but also previously the US and also Germany and Spain. And the Complex Systems Institute here in Paris, CNRS, and École Polytechnique, among other things. And all that has now taken a new sense here at Manchester Met. All right, so let me, right off the bat, I want to cite, I want to make clear that I'm not, me and in fact Martin Amos, who is here, the head of the IRC, the Informatics Research Center. We are not alone in this endeavor to take biology as a source of inspiration And I want to cite no less than Harvard, who 10 years ago created a new institute called the VIS Institute, of course, anonymous, very well funded. And so let me read their manifesto, the most important aspect, which is we are now beginning to understand enough about how nature builds, controls, and manufactures, so that entirely new engineering principles will be discovered. All right? We must harness these insights to create biologically inspired materials and devices that will revolutionize healthcare. In their case, it's focused on healthcare, biomedical engineering, and with this strange loop of bio-inspired biomedical engineering, right? But I should add, this is me here, that it also could be very well applied and is applied to swarm robotics and socio-technical networks and all other kinds of topics outside of healthcare. So I also want to bring as another witness, if you wish, to plead the case of unconventional computing or novel computing, Professor Susan Stepney from New York, a very good colleague and friend of mine, who had a very nice keynote, and in fact, I think a cover article in this European magazines a few years ago about extending the famous quip by Stanislav Ula, according to which non-linear systems is like talking about non-elephant biology because it's everything. Almost everything is non-linear. So why don't we do the same with non-Turing computing? Is also talking about so many other different ways of computing than the traditional Turing machine as brilliant genius it was and is the basis of all our classical, our computers today. There are so many other ways to compute that could be inspired again by physics, biology, chemistry, and so you have DNA computing to cite, for example, this one, and everything that is neural networks, genetic algorithms, and colony optimization, all those disciplines that are inspired by biological phenomena. And all that belongs to complex systems. All that is about looking at complex systems in nature, self-organization, and this is how I will start my presentation. I will set the stage briefly and remind you, if you don't know already, or tell you what is generally understood to be complex systems and their main properties of decentralization, emergence, and self-organization. And then I will move on and talk about morphogenesis and other topics. So, complex systems are everywhere around us. Nature is replete with them at all levels of organization, from molecules to ecosystems via, obviously, organisms, whether plants, animals, or the animal-human species, and the societies, the collective constructions that they make, and in our case, of course, technology. All that creates this infinite hierarchy of systems and systems, where at every level, the main properties are self-organization, in the sense that large sets of agents interact only locally, right? Emergence, so their simple individual behaviors create collectively a complex emergent behavior. So emergence is also a hotly debated concept in philosophy. And decentralization was the last one. So there is no grand architect or blueprint set in advance. So let's go through, I'm going to take you on a very quick journey through the land of complex systems. And we can describe complex systems in terms of big families of phenomena and models. Typically, for example, this is of course not canonical, this is not the only way to try to categorize complex systems in a vast field of complex systems. But typically you would have pattern formation, this is a big one, whether physical or biological. You have, of course, a well-known physical pattern formation with convection cells, hot and cold fluids, forming those whirls and creating spots and stripes, but also very different pattern formation here. Animal codes, which can be abstracted and simplified into this model, this discretized version of the Turing model, so Turing is here too. But in this was, of course, his other, one of his many streak of genius, the morphogenesis that he made clear that you would have order emerging out of initial randomness by amplification and fluctuation. You could have spots and stripes, so activation and deletion, or reaction diffusion models. Anyway, so this could explain those animal stripes and spots. Other big families, synchronization, generally speaking, and of course, not a small instance of this family is our brain, where you have 100 billion neurons synchronizing their electrical signals in very complex ways to form patterns of activity, which are our thoughts, our emotions, our actions, our perception, and so on. And swarm intelligence generally refers to those collective, to those insect colonies who remarkably together create those superstructures which are bigger than them naturally, so trails in the case of ants, by laying down pheromone and sniffing pheromones, so amplifying those trails, so and together in fact computing something bigger than each individual. So this is why it's called swarm intelligence, because they, in fact, they are not computing the shortest path. Even though no ant has an idea of what short path is. And two other families, and collective motion is another big one, also studied a lot by physicists, statistical mechanics, statistical physics, so how animals, birds, cattle, fish, cluster together and move together in flocks and herds and schools. And again, every time I show you those examples, I also show you a very simple model. What is perhaps, of course, an extremely simplified model. In that case, mostly using NetLogo, which is a very nice educational platform for complex systems. And at this point, I was wondering, sorry to interrupt, if we can dim the lights in front of the screen. Is there a way to do this? So you hit it right. I don't know where it is, sorry.
Try to press the button.
Sure. Which one?
Yeah, this one. No.
Shedding a lot of light on my door. Thank you, Jan. That's better. Thank you so much. Excuse me. Anyway, so yes, in the meantime, the slides move, but I wanted to finish with this. So anyway, so yes, flocking, collective motion, big topic too. Those fantastic flocks of birds. How do they do that? And so I was saying every time there is a simple model which can capture the essential mechanisms. So in that case, for example, birds are dots and they obey separation, alignment, and cohesion principles, and that's it. That's all you need to kind of reproduce this collective behavior. You don't need to dive into the nervous system of the birds and so on. You can abstract, you can model every time. So very important thing, attitude to have, keep in mind. Modeling is about abstracting and simplifying the agents of the phenomenon. And of course, another category, diffusion and networks, and in that category, also a vast domain of all kinds of human-caused systems, such as urban morphogenesis or, of course, social networks. Big topics in the past 15, 20 years, complex, skill-free, small world networks of relationships and so on. Good. Anyway, so that was again, a kind of a very fast, sweeping overview of complex systems, just to try to give you a better idea of this domain. And so here is the brochure that you can get at the entrance of the complex system spark, and also where molecules give rise to cells that give rise to organisms and their nervous system, organisms in turn giving rise to all those collective behaviors, and this organism, the human species here, with its technology giving rise to all those super overarching societal phenomena that fascinate and worry us so much, of course, markets and the web and populations and many other phenomena. So now, all this being said, on this vast stage of complex systems, I showed you so many different things. I want to focus here on a subcategory, those complex systems that exhibit an architecture, that exhibits some non-trivial structure made of modules, of parts, some heterogeneity. Those are, in my view, the most intriguing. I mean, the one I prefer focusing on, and here they are. so from this earlier panorama of complex systems, I'm now focusing on those here, because why those? Because these exhibit the possibility of combining self-organization and an elaborate architecture. This is not the case, for example, bird flocks, as fascinating as they are, they don't have an architecture. They are very amorphous and they're fluid everywhere. Spots and stripes are very interesting too, but precisely this is what, in fact, Turing himself is alleged to have realized, that he certainly proposed an interesting model of stripes, pattern formation, but this is not morphogenesis in the strong sense of the term. Morphogenesis, the creation of form. So he is alleged to have said that he had the stripes, but he didn't have the zebra around the stripes, indeed. So morphogenesis, I want to insist on to insert a wedge here to kind of separate more clearly pattern formation from morphogenesis. And so this is, of course, the epitome of that are multicellular organisms. When you think that this body plan and this exquisite detail of all those organs and parts and so on arises from one fertilized egg. And to some extent also the construction of termites are very fascinating too. No termite is in charge of the plan of the mound, and yet it is so sophisticated. So in fact, I just started talking about those two other families, morphogenesis in the biological sense here, and collective construction. So these are, in my view, these are really standing out from the rest of the vast complex systems field, because again of this incredible elaborate architecture that they are creating spontaneously. And again, so a termite mound, for example, is also like an anatomy. If you cut it open, this is a model, plastic model, I think. If you cut it open, you see an incredible diversity of structures, chambers and corridors, hallways, chimneys, gardens, and so on. And there is no termite in chief that is directly on it. And same here, you have also an incredible array of various parts and so on. Okay, so this being said, what I want to do every time is, let me go back just a second. So I want to talk about this domain here of architectures without architects. So self-organized systems that look like they were designed but are not. All right. So sometimes sometimes a political or religiously heavy notion. Anyway, and I want to show you in this case one study, an important one, which I collaborated to. In fact, in that case, I was the leader of this model with a PhD student, René. So this is about modeling such a morphogenetic complex system, in that case, embryogenesis. And it is about observing embryogenesis under the microscope, observing the development of an animal, and then I was leading the modeling part of it, which is here. On the other hand, there was a big team in the lab and European partners doing an analysis of these images, automated image processing of those movies to extract the positions of cells, and then we needed to think about what mechanisms would be able to reproduce by simulation here on the right this type of phenomenon. So here on the left you have the measured embryo. Here on the right you have the simulated embryo. You understand? So here, and I'm now going to go through those steps here, which are here, smaller size. So first I'm focusing here on measuring the embryo, so measuring, automatically detecting the cells inside this embryo developing under a microscope, which is a huge endeavor. It's a complicated image processing flow of various modules of filtering and segmentation, and so it was the work of a big team of mathematicians and computer scientists, two big European projects of the past 10 years. Anyway, so in the end what you have, you can superimpose those automatically detected cells with the original movie. This is one direction, right? This is not about modeling here. It's about measuring, detecting, analyzing, okay? And then the next step is this one. This is the one here at the top. This is the core of the model. Now this is necessarily more abstract, necessarily simplified model of embryogenesis. It doesn't need to capture everything. And indeed, like the birds earlier, which were dots flying around, the cells here, well, they're also dots, but a little more complicated than the birds, perhaps. They also have forces. I'm not going to explain everything in detail, obviously, but they have forces, protrusion and elastic-like adhesion forces, intercalation, things like that. And each of them also contains a GRN, a genetic regulatory network. So everything you see here in this slide, you have a visual summary of the model. The model, so the mechanics behind embryogenesis, or that could explain some of the early stages of embryogenesis. Again, the ambition is not to explain everything. That's beyond the capacity we have today. But let me just spend a few more seconds on this. So you see that this cell is not represented as at a molecular level. It would be, of course, we could try to represent the membrane as the bilipid layer of molecules, and so, but no, it would be way too expensive computationally. So we have to take shortcuts. But of course, there is not just one cell like that, you have hundreds of them. So this is why simplification in that case is enough to, and here you have a simulated embryo, right? So we are here on the right-hand side of this flow, the simulated embryo, is enough to account for, to some extent, of course, for example, this episode of the zebrafish development, which is called epibody, where this layer of cell is sliding along the yolk and trying to envelope the yolk. So I'm just assuming a few elementary forces here, imagining that cells behave a bit like Newtonian objects, which they don't really, but of course, again, we need those simplifications. And then by intercalation into each other, they create this collective motion. So again, there is an emergent collective motion phenomenon out of individual cell behavior motivated by a few forces that we chose or modeled carefully. Right? So this is how the kind of simulations we create. I know, but of course, remember, this is an exercise in computational modeling. This is the biological modeling side of things. This is not robots and new technology and funny things. This is trying to help biologists understand their object. Okay, so we need to be as realistic as possible. We are not here in the business of creating funny, colorful movies or computer graphics for the fun of it. But we need here that our simulation on the right-hand side, it's another type of simulation here, is in fact consistent with what was measured, which is here on the left-hand side, except those two things are different from what you saw before, just other examples of simulated embryo on the right and measured embryo on the left. Okay, and so here is the crucial part, in fact, after all this effort, is trying to optimize or to tune the parameters of the model. And to do this, we simulate embryos many, many times over with different parameters, because the model is, of course, full of parameters. And so we need to search parameter space. This is also a very general methodology that you apply to all those complex systems models, because complex systems models definitely have a great number of parameters, and you need to tune them so that your model will match the data as well as possible. All right. So this is the final step is the fitness, or the how the simulation can indeed reflect the measured data by superimposing, for example, vectors. And here we find, in this case, it's an example, one parameter here seems to have a preferred value, or a best value, or most realistic, or least unrealistic value. All right. Okay, so that was computational biology as an example of architectures without architects, how to model and understand them. Let me switch to something quite opposite, another subdomain of complex systems here that I found also very fascinating. Architects overtaken by their architectures. So how design systems, so quite the opposite, became suddenly complex. And I will show you also one study that illustrates this family. It's geographical morphogenesis. So what am I talking about here? I'm talking about the bottom part of this brochure, this kind of panoramic view. The bottom part being all those societal phenomena. And those big societal phenomena that we're so fascinated with and intrigued, or again, that we are worried about, they all arise from very rigidly designed artifacts. Cities arise from buildings. Internet and web arise from computers. And markets and economy arise from companies, institutions which are also much more rigidly organized inside. And so all those big phenomena here are the result of millions of those, billions of those, interacting together and forming something that completely supersedes us. So at that level, at that high level, no one is in charge anymore. No one has planned entire cities, or even though it has been tried in the past but not very successfully. No one has planned the entire internet, no one has planned entire economies and so on. So at this large scale, you have a de facto complexification of organizations via socio-technical networks, because we have now ubiquitous ICT capabilities, ICT computing and communication capabilities, connecting people and infrastructure in unprecedented ways, and giving rise to those complex socio-technical ecosystems composed of a multitude of human users and computing devices. So there's an explosion in size, in complexity in all domains of society, all right? And in fact, there is a shift from a centralized oligarchy of providers top-down to a much more bottom-up. So in fact, it has accelerated. It has become even more mind-boggling than before. So having made this observation, we can try again here too. I showed you briefly an example of a study. So again, this is a study trying to analyze, simplify again, not an embryo, but this time a city. So but very much along the same principles, how a city grows. In this case, again, yet again, an abstraction is required. So houses are dots and roads are edges of a network. So it's a network on top of a grid, by the way. It's a network on top of a cellular automata. And the research question here is it possible to reproduce patterns of urban form? So urban development, taking into account constraints about transportation and services and things like that. So this was the goal. I contributed to this study that was headed by geographers in Paris. And again, this is only one slide about the model, so can reminiscent a bit of embryogenesis, but of course the goal here is very different. We also simulate, these are artificial simulations of possible city morphogenesis using cellular automata and using also the growth of networks. And here you see you can give rise to very different patterns, in fact, web architecture patterns, depending on parameters. Of course, I will not explain the details of this model, just to show you that the core of the model is here. It's about an energy function that has different constraints in it, about distance to services, distance to roads, and so on. So every local patch of geography is making a decision as to building a house or extending a road. And this decision is based on beneficial energy, beneficial gain in energy or not. This is really the idea there. Okay. And also to show you, it was not just like the embryogenesis model, it was not in vain. We're not just producing colorful movies. We try also to link them with reality and real data. It was also applied to in a minimal way. There was no impactful application in the real world, but it was an attempt to show that this kind of model I just showed you could also take as input real data from, for example, a district from a suburb of Paris, and try to produce out of this existing map some recommendations, some outcome on how businesses should be best positioned and things like that. So just to give you an idea, this is a bit the same spirit as embryogenesis, except in embryogenesis we were not giving recommendation to the embryo on how to grow. but also trying to match the real phenomena. All right. Now that I have talked about complex systems very generally, after that I talked about those complex systems exhibiting strong architecture, morphology, but without any architect, and I give you the example of Morphogenesis. I also talked about a bit the opposite, of those systems becoming organic at a higher level, but coming from very rigidly designed artifacts. I want to show that these two categories, in fact, converge to a middle ground that I call morphogenetic engineering. It's a field that I founded a few years ago, several years ago with our colleagues, and tries to capture those two sides and unify them and try to reconcile, in the end, architecture and self-organization. So this is can we reconcile those apparently antagonistic concepts of things being having a very specific structure, but things also being completely spontaneous in their emergence. Is it possible to have both, the best of both worlds? And I think it is. And this is what I call morphogenetic engineering. So on the one hand, again, we have those rigidly engineered systems showing no self-organization, or very little, or if they do, it's a nuisance. We don't want them to be self-organized. On the other hand, we have those natural systems that have no architecture. So flocks and stripes and spots and clusters and waves, all right? So this is completely opposite on the spectrum of going from architecture to self-organization. And I have gone over those two middle categories. This was the green blob. These were the architectures without architect. This was the blue blob. These were the super structures. And in fact, these come from those, but the elements inside them are loaded with more information. And vice versa here, these come from those, as we know, but in that case, those rigidly designed artifacts are multiplied by millions and start to spread out in non-rigid ways. Anyway, in the middle is morphogenetic engineering. Let me now define it more precisely. And shameless plug from the book that was published a few years ago. Morphogenetic engineering is about designing the agents of self-organized architectures, not the architectures directly. So after studying the natural systems, can we do the same in artificial systems? It brings a new focus in complex systems engineering. Complex systems engineering, another oxymoron. It explores the artificial design and implementation of decentralized systems capable of developing elaborate heterogeneous morphologies without central planning and without an external lead. And it belongs to what could be called generally engineering and control of self-organization, which has seen in the recent years a flurry of creation of new disciplines that all sound a bit similar to morphogenetic engineering, with different focus, different highlights, but I must mention guided self-organization, for example, is another field launched by a colleague. Organic computing was a German initiative. Programmable matter, also steering complex systems. So you can see that a lot of those phrases, those names of computing disciplines, contain in them this paradox that they are trying to solve. Steering on the one hand, complex systems on the other. Guided on the one hand, self-organization on the other, and so on. Right. It's all about the same kind of trying to put things together. All right. So I will finish my talk by a bit like before, by showing a few studies that relate to morphogenetic engineering. And how am I doing in terms of time, Craig, at this point?
10 minutes.
10 minutes? Okay, that's good, thank you. So, what have I done in morphogenetic engineering? I want to show you one study about artificial life creatures, another one about swarm robotics, and then there will be two others here showing you later. So, this is what really started a lot of things now almost 10 years ago, 8 to 10 years ago. So this was, I decided to have fun and try to do artificial development in the computer. So just imagine, in that case, not being bound to realism, to a real organism like the study I showed you earlier. This is called artificial life. This is the epitome of, the essence of artificial life, not epitome, sorry, but typical of artificial life, where you muse about life as it could be. Life as it could be in the computer. So this model here, this 2D model, was trying to capture the essential principles of biological morphogenesis, which are cell proliferation and patterning. So cell proliferation, division, and patterning via gradients. So the cells are communicating with each other and exchanging positional information. They are counting themselves 1, 2, 3, 4, 5, without any lead from any centralized control. And they are giving rise to this body plan here, this creature. And what is important is that every cell contains the same program, which this is a bit like the model of embryogenesis with forces and gene regulatory network. This box contains about the same kind of principles. Every cell is going to follow a certain behavior depending on what it receives from the neighboring cells. And then together they give rise to this specific form. So it is not flocking, it is not random clustering, it is not just forming a blob. It is creating a specific shape with a tail and fingers or whatever it is. Okay. And so every time you run this simulation, you get the same result, but it's not exactly the same either, because there is still a bit of randomness inside. There is a bit of course, there is noise, there is movement, so you won't have the same number of cells or the exact same position, but you will have the same body plan. So I was trying with this to capture the essence of self-organized architecture with this artificial 2D creature, and then later on we expanded that, worked to 3D with a student in Spain. I will cite all my acknowledgements at the end, of course. And then, so here in this case, same principles of gradients, crossing the swarm of cells, also elementary physics, elastic forces between the cells. And then growth from there by patterning, because the cell mass is segmenting itself in different areas. That is very important. You need to have heterogeneity is key in this case. You need to have a heterogeneous set of agents so that some of them can start growing a leg, but some of them will do something else, which is very different from flocking birds. The flocking birds are homogeneous. All the birds are doing the same thing. There is no birds more to the west are going to flock in that way, and the birds to the east of the flock are going to flock differently. No, they are all the same species, and so they do the same thing. So here it's as if you had a multi-bird species flocking, if you wish, trying to connect and if I contrast those systems again. All right, so this was what we did, and then we had fun putting this developmental creature into an artificial physics environment, so physics engine, and see what happens. So we animate it, so it hatches, and then it is animated in the world and can do different things. It can walk, it can kick a ball, it can climb stairs. We also studied a bit its anatomy inside. This is the skeleton inside, and so we try to mutate the poor thing, and so it's only skeleton, or vice versa, it's only muscle but no skeleton, and see what happens. So we can, of course, tweak the artificial genome in any way we want. And so all that seems, I want to insist on, I think, how important this kind of exercise is, even though it is disconnected, it seems to be disconnected from real biology. Obviously, this is really free creation of fictitious biology in an artificial world, and yet doing that, I think, and I'm not the only one, the whole artificial life community thinks the same, that it is essential to then come back to the real biology and understand it better with those new ideas that you have. I want to insist on that, if I can. I like to use, for example, the example of the complex plane, C. It was surely a surprise when you were, I don't know, 11th grade or 12th grade to meet the complex plane for the first time. And what is this little I, whose square is -1? Strange, fictitious numbers that have no reality. And yet, thanks to this bigger space than R, the line of real numbers, then you can solve many things. You can go back and solve real problems thanks to this foray into imaginary number. Well, it's the same idea here. Thanks to this foray to imaginary biology, you can come back to the real biology and address it with new ideas and solve problems in that way. Anyway, but this is not what I'm doing right here. Yes, just to show you the end of this study where you can mutate the genome and then create all these different and kind of do a mimicking a bit evolution. All those are simulations. So what for, besides what I just said, perhaps helping us modeling real biology better? Well, there is another exit door which is completely at the opposite, which is applying what I showed you to robots, for example. So those colored dots could be robots, small robots that could self-organize and form a bigger structure than themselves. Maybe the transformer way, if you wish, or I don't know. So swarm robotics, but morphogenetic swarm robotics. So again, you see that everything I do is prefixed with morphogenetic in this category. Morphogenetic swarm robotics, where first of all, collective robotics is a big field of investigation. The benefits of decentralizing robots into a myriad, distributing them into a myriad of small robots are very well known. So you have robustness and local communication and things like that. And if you fail, never mind, because you have all the others that can take over. But at the same time, it's very promising. It's very difficult to control, of course. Instead of one big clunky robot, you have dozens, maybe hundreds of them. And so it is a challenge to control them. And this is where I hope to contribute to this field, bringing these ideas about morphogenetic engineering into collective robotics to make those robots self-assemble, very much like those artificial cells I showed you earlier. But of course, we need to do that. We need to solve many robotic specific problems that were not the case in the color dots. So, but the same idea of forces and genes that would, or forces here and gradient there, that would together make those robots self-assemble and disassemble and reassemble again into superorganisms. And I want to show the first very preliminary steps of this here. So I had an intern this month of August here at Manchester Met, Philip coming from the University of California, who started with those sci-swarm robots, which we are acquiring from the University of York, from John Timmis. So here are the robots here, and here are the few first robots that we got. And so Philip was experimenting with making them simulate elastic forces. So this is a virtual elastic forces between the robots, which is of course the prelude to a bigger swarm and self-organization based on that. Okay? Good. All right. And the end of my talk, I might skip the last one. I want to talk about two other studies in morphogenetic engineering. One is synthetic biology and one is technosocial networks. This one is probably the most mature. So just to show you that we can, in fact, now this illustrates what I said earlier about coming back to the biology with new ideas about morphogenesis that we want to then suggest to the biology. And we will not do that to any animal embryo, obviously, but we can do that with bacterial mats. In that case, this connects very nicely with what is called today synthetic biology, which is a very active and growing, fast-growing domain about rebuilding, if you wish, or tweaking with the genome of existing cells to make them achieve useful functions for us, such as typically transform chemicals, create food, energy, and so on. But so far, this field has been mostly looking at a well-mixed soup, the model of, and what I try to contribute to is the next generation of synthetic biology, at least modestly in the computer for now, but with the intention of one day making it into better dishes. And about, again, can we dream, can we fantasize about perhaps harnessing those bacteria to make them grow, construct things that they are not used to doing, but we have modified them, of course, in a way that they will do morphogenetic engineering for us. All right, it's a bit like robots, but in that case, robots cannot divide, cannot proliferate, cannot stick to each other. Bacteria can. So why not use them? And perhaps one day we'll have that, and they will fossilize and give us naturally grown buildings, something like that, for example. So this study was the first step in simulating. So don't worry, there was no wet lab experiment, nothing. No bacteria escaped. It was all in silico, but with Jonathan, we programmed this platform to make bacteria again self-organized. And so again, this is the same idea that you have one code or one script inside every individual field, which is following rules of diffusion and proliferation and differentiation into different types and so on. And what we obtained in the end was that. So we have the simulations that were published in synthetic biology about showing a proof of concept, showing the feasibility, perhaps, of modifying the genomes of bacteria such that they are going to grow specific shapes, and not just a film of all of them spread evenly. We don't want them to be spread evenly. We want them to spread in specific ways: 3-arm, 4-arm, T-shape, L-shape, and things like that. So again, it's about reprogramming, in that case, the agents such that they would create the architecture that you wish to see. Okay. All right. So I will skip this last one because yes, I think I've reached my. This was more the techno-social aspect, of very abstract, and in fact, it's not really mature yet. And I want to acknowledge my collaborators and also cite my current and future collaborators after this. So a lot of people have contributed to studies. I also highlighted in color the students who are really the master of the code. So Julien in Mecagen, Just in Erbmorph, Carlos in MapDevo, Jean-Marc Montagnier, postdoc, and Philippe, DevoBot, the beginnings of DevoBot, Jonathan Pascali, Symbiotic, and those two here probably, which I skipped, sorry. And I want to cite, of course, that in this domain, in fact, of socio-technical networks, we are starting now with Emma Morling and also Bruce Edmonds at the business school, a PhD with Nicolas, who is here, new PhD students, who will do work along those lines. And then, the robotic topic, I need to find a student, but of course I have my colleagues at York, who are really, of course, very close to that. And then, yes, I want to mention MapDevo. So we have with Matthew Crossley and our RA, Carlos Herrera. We are also trying to expand this into more complex games environments and see how the culture is doing in a complicated world. So anyway, thank you for your attention.
Thanks very much. 45 minutes? Yeah, very good. Colleagues, any questions for me?
How many?
You get one.
I enjoyed very much your talk, and in particular that place where you mentioned the essential principles of morphogenetic engineering, mainly proliferation and patterning, right?
Patterning and mechanical forces and division, yes.
So, do you consider mechanical forces as an intermediate between proliferation and patterning, or it's just some built on the... I mean, my question is actually, biologists do not know what makes individual cells pattern. And that's the main question in biology until now. And probably it will be resolved unless IT people will have, right? So my question is morphogenesis, from the point of view of evolution, essential thing, just optional? In other words, why start out growing in the world like just concepts, right? Or colonial organisms.
Yeah, homogeneous.
Any morphogenesis, which is really similar because morphogenesis needs a lot of evolutionary energy to create, right?
Yeah, that's a good. I mean, you are talking about why the division of labor occurs spontaneously.
Yes.
Why those assemblies of cells, which perhaps a long time ago were homogeneous, in fact, bacterial mats are pretty much homogeneous. Cancer has a complicated morphogenesis inside. It's not that homogeneous. But anyway, yes, why did evolution favor, encourage division of labor, differentiation, regionalization of the assembly into different parts that could do different things? It's a very good question. I don't know, but it probably had an evolutionary advantage because, perhaps the fact that different functions could be different shapes or components, parts, could arise at different places was useful for the whole organism to be able to perform in a complex environment.
I'll ask another question with wine, okay?
Yeah, only one.
I'll ask another question with wine.
Wine. Yes. I love that question.
Questions with wine.
Very much.
Any other questions, colleagues, before we go out to the wine?
Oh, you shouldn't have mentioned the wine because everyone wants
No?
Go on.
So I think that's it, René. But thanks ever so much for coming to
Okay.
He's going to ask over wine.
Over wine, okay, so everything is supposed to go
We'd like to thank you, René, ever so much for coming to
thank you.
You're very welcome. And we'd like to get more people like you.
Oh.
So thanks again, René.
Thank you, Chris.